Aging isn't just a matter of worn-out organs and tired mitochondria, it's also a story written at the level of gene regulation. As we grow older, our epigenome, the layer of biochemical marks that determines which genes get switched on or off, begins to drift. Genes meant to stay quiet are suddenly active, while others critical for repair, detoxification, and longevity get silenced. This phenomenon, sometimes called epigenetic drift, is one of the key hallmarks of aging. And now, in a surprising twist of molecular fate, researchers are beginning to ask, could psilocybin, the compound responsible for the effects of magic mushrooms, help restore this fading epigenetic script? It sounds fantastical, but the science is catching up. When psilocybin enters the body, it's converted into psilocin, a compound that binds to 5-HT2A serotonin receptors, which are found not just in neurons, but across multiple cell types. This receptor activation sets off a complex cascade of intracellular signals, many of which reach the nucleus, the control center of the cell, where the epigenome lives. While most attention has focused on psilocybin's impact on mood and neuroplasticity, mounting evidence suggests its reach may go even deeper. It may alter the chemical tags on DNA and histones that control gene expression. One of the main ways the epigenome changes with age is through DNA methylation. This is where methyl groups are added to cytosine bases, especially in regions called CPG islands near gene promoters. In young, healthy cells, methylation patterns are tightly regulated. But as we age, we lose methylation in some places and gain it in others, often silencing longevity-promoting genes while activating inflammatory or senescence-related ones. Interestingly, some preliminary animal studies and in vitro experiments show that psychedelics, including psilocybin, may reshape these methylation patterns, possibly reversing some of the epigenetic noise that accumulates with age. One study on related serotonergic psychedelics found changes in expression of DNA methyltransferases, DNMTs, the enzymes that add those crucial methyl groups to DNA. Another showed modulation of TT enzymes, which remove methyl groups, allowing silenced genes to be expressed again. If psilocybin influences these enzymes even subtly, it may be capable of resetting key aspects of the epigenetic aging clock, a molecular measure of how old your cells think they are. But it's not just methylation. Histone modifications, the chemical tweaks that determine how tightly DNA is wrapped around its protein scaffolding, also appear to be influenced by psychedelic exposure. Psilocybin-induced neuronal activity has been linked to increased expression of genes tied to histone acetylation, a modification typically associated with more open, transcriptionally active DNA. Translation? The cell becomes more capable of expressing plasticity-related and repair-related genes, essential in tissues like the brain, where regeneration slows dramatically with age. These changes may not be limited to the brain. Peripheral tissues, including immune cells and even intestinal epithelial cells, express serotonin receptors. Psilocybin could influence their epigenetic machinery too, helping reduce systemic inflammation, improve barrier function, and enhance resilience against stress, hallmarks of a younger, more responsive physiology. What's especially compelling is that these epigenetic effects may explain the long-lasting impact of a single psilocybin dose. People treated for depression, addiction, or anxiety often report benefits that last months after just one guided session. That's not typical for drugs that merely tweak neurotransmitters. Those wear off in hours. But when gene expression is reprogrammed at the epigenetic level, the changes can endure for weeks, months, or longer. This suggests that psychedelics may not just lift the fog temporarily. They may rebuild the biochemical scaffolding that supports mood, cognition, and recovery. Of course, we're still in the early days. Direct human studies measuring methylation clocks before and after psilocybin treatment are sparse, but likely on the horizon. The epigenome is complex and context-dependent, and not all changes are beneficial. But the idea that a mushroom-derived molecule could reset aging cells at the level of gene expression isn't as wild as it once seemed. In a way, it's poetic. Aging writes in slow, silent edits, adding methyl groups here, tightening chromatin there, closing down the body's once fluid potential. Psilocybin, long known for expanding consciousness, may also expand the cell's sense of possibility, reopening closed genes, silencing those that shouldn't be shouting, and gently coaxing the genome to remember how to be young again. So while the phrase psychedelic anti-aging therapy may still sound fringe, the evidence is tilting toward a serious scientific possibility. not by hacking DNA or injecting stem cells, but by listening to the language of methyl groups, histones, and serotonin receptors and whispering, reset.